Hello, everyone. Thank you for being here with me today. My name is Carole Estoppey. I'm currently a principal scientist at ICNOS Sciences. And today I would like to tell you about BEAT 2.0, which is ICNOS' next generation multi-specific antibody platform. So ICNOS Sciences is a clinical stage biotechnology company, which is at the forefront of innovation in oncology. It is a fully integrated biotech with expertise in biologics from discovery to antibody engineering to CMC to clinical development, with a global footprint of about more than 200 colleagues in US and in Switzerland. And this is where I am based, in Switzerland at the moment. Our approach is to focus on immuno-oncology with multi-specific immune and non-immune cell engager antibodies. And most of that is based on our proprietary BEAT antibody engineering platform, which represents our discovery engine for innovation, whereby we are generating next generation multi-specific immune and non-immune cell engager antibodies that can engage multiple targets simultaneously. And I will tell you more about our pipeline in the next slide. ICNOS is currently advancing a very differentiated pipeline with potential first and best-in-class assets. Our two most advanced assets in terms of bispecific antibodies are ISB 1342. That's a CD38 CD3 T-cell engager that's currently in phase 1 for relapsed refractory multiple myeloma. The second one is ISB 1302. That's an HER2 CD3 T-cell engager, which is currently also in phase 1 for metastatic HER2-positive breast cancer. We are also advancing bispecific antibodies at an earlier stage. Two of them are currently in pre-IND phase. That's ISB 1908, which is also a CD3 CD38 T-cell engager. And another one is 1909. That's another type of T-cell engager. And ISB 1442, which is a CD38, CD47 BEAT bispecific antibody, which we're developing for hematological malignancies. We also have two monoclonal antibodies currently in development at different stages for autoimmune disorders. That's ISB 830, our OX40 antagonist, which has completed phase 2B, and also ISB 880, which is an IL-1RAP antagonist monoclonal antibodies for autoimmune diseases. So now moving on to bispecific antibodies. Making bispecific antibodies is really a tale of dismantling and reassembling antibodies. So this is what an antibody looks like with two same heavy chains and two light chains attached to each heavy chain. And if you want to make one that will bind target 1 and 2, as is depicted at the right of this slide, you can imagine that if you just co-transfect in the same cell, one of heavy chain 1, heavy chain 2, and the two different light chain, there's a lot of different products that can assemble because the cells will be able to assemble any heavy chain with any light chain and any heavy chain with another heavy chain. And so there are really challenges to get this particular assemblies of bispecific antibody. So in order to do that, you really need the optimal assembly of 4 different chains, and you need to drive that. And your first step in making a bispecific antibody is to assemble the heavy chains via heavy chain technology. And at ICNOS, we do this with what we call the BEAT interface, and I will tell you more about that in a couple of slides. Once you have your 2 heavy chains properly assembled into a heterodimer, you also want to assemble the light chains via light chain technology. And we've done this in 2 ways at ICNOS. The first way was to bypass this problem by using a Fab on one side and an SCFV on the other. And then in a second step, we decided to move to a common light chain technology. And I will tell you more about that later. And then once you have that technology in place, you really would like to be able to move on above bispecific formats into trispecific antibodies by using the same building blocks you've had for the bispecific antibodies. And not a lot of technologies out there actually allow to do that. And our technology is one of them. So the BEAT platform actually focuses on 4 different pillars. The first one is a wide design space and plenty of room for optimization, whereby actually we can explore the design space covering different affinities of the different building blocks. We can target different epitopes of a similar antigen, and we can look at lots of different valency and formats in order to really get that flexibility to hit the right biology with the right potency and the right specificity. We like to do that in a format that's very close to the natural antibody format that we believe will enhance the stability and enable high concentration, high yields, very little product-related impurities, and enhance pharmacokinetics. Based on that, we think we can target broad therapeutic applications in oncology with an ability to develop different categories of immune cell engagers, both for hematological cancers, but also for solid tumors. And with this flexibility, we can also unlock new biology with an ability to open therapeutic window, have better specificity, enhance or silence the FC in different ways to really get those antibody drug candidates to be fully optimized. And what the BEAT technology is initially based on is a heavy chain technology, which is a leading proprietary technology. And we call it the BEAT interface. The BEAT stands for Biospecific Engagement of Antibodies based on the T-cell receptor. This interface has been published in 2017 in JBC by Skegro et al. Feel free to go have a look at that paper for all of the details how it was done. And then we published a further enhancement of this interface in the same journal in 2020 with the Stutz et al. Paper. The way this interface works is that we have used residues issued from a naturally occurring heterodimer, that's the TCR alpha beta heterodimer, which has a structure that closely resembles in 3D to the antibody FC. We have taken the specific residues that are involved in the heterodimerization of the TCR alpha and beta, and we have grafted them onto the antibody FC in the CH3 domain. You can see the residues that we've grafted onto what we call arm A are depicted in blue here, and the residues we have grafted onto arm B are depicted in red onto this structure. This TCR interface that we've grafted onto the antibody FC drives very efficient heterodimerization of the antibody. It is buried within the contact between the two chains, making it a potential best-in-class technology. And this technology is efficient enough for us to be able to efficiently, routinely prepare bi- and multi-specific antibodies using just a single step of protein purification and reaching excellent purities after this step. Now, this is about the BEAT interface. And we use this BEAT interface to make two different generations of BEAT molecules. Our first generation, which we call BEAT 1.0, was making use of a Fab scFv format. As you can imagine, one way to bypass the light chain pairing issue is to have one arm using a Fab format and the second arm using an scFv. scFv means single chain F fragment variable, and that's just an assembly of a VH and a VL via a flexible linker, which allows to reconstitute the binding site for your target antigen without the need for a separate light chain. It's a very versatile and quick way to make bispecific antibodies, and this has allowed us to bring 1302 and 1342 to the clinic. And you also have a 1.0, which can be optimized into 2+1 format, as is seen here. But what we have noticed is that we wanted more space for optimization and engineering of the different arms, and also because of the scFv format, some challenges in CMC for high concentration formulation and high dosing indications could be challenging. And this is why we decided to move on to BEAT 2.0. As I briefly said before, BEAT 2.0 makes use of a common light chain, which is depicted here in black. It essentially means that the two binders, which will bind two different targets, are varied in the VH, but carry the same light chain, meaning you bypass the issue of common light chain pairing. This Fab format allows a better optimization. We can look more easily into different affinities and explore the epitope. And it enables CMC for milligram per kilogram dosing indication and eases high concentration formulation. And now, since your building blocks all carry a common light chain, you can also go to tri-specific antibodies and multi-specific antibodies. This is also a very versatile and plug-and-play approach, but it does require specific discovery platforms that are built around a common light chain, which we now have enabled at ICNOS. So once you build your common light chain binders through your discovery engine, you can imagine that there is no limit to the combinations you can achieve in a very plug-and-play approach. You can now engage T cell, NK cell, or macrophages and different tumor antigen in a variety of formats in what we call 1+1, 2+1, or tri-specific formats. And this is really the beauty of this technology, is that it allows us to explore the design space in a very broad fashion. Now, in summary, the Beat 2.0 and Treat 2.0 is a plug-and-play approach to design multi-specific formats that could address novel biology. We have a natural IgG format with the natural TCR interface grafted, which is not solvent exposed and thus potentially best in class. And we also have a natural heavy and light chain pairing through the use of the common light chain technology. This enables standard antibody CMC processes and easy process development with high purity after a single chromatography step with the protein A avidity method. It's a plug-and-play approach that it's enabled by internal discovery platforms, and it is readily amenable for the design of not only bispecific, but tri-specific and multi-specific antibodies, with the possibility of having prolonged half-life. Now, moving on to show you a bit more data about the purification of these beads, I wanted to take you through an example of the purification of a bispecific antibodies in the Beat 2.0 format. But before that, I also wanted to tell you how we use the Twist Bioscience DNA synthesis to make this plug-and-play approach even more efficient. So the use of Twist Bioscience's DNA has really made it easy for us to enable this plug-and-play approach. And we use Twist DNA in two different approaches. The first one is when we order cloned genes, including scale-up and custom cloned into our own vector. This is really efficient, and it means less hands-on time for our laboratory staff, and they're really happy about it. And it's also very cost and time efficient. But sometimes the long chain that carry 2 VHCH1 repeats are challenging to synthesize and to clone into custom vectors. So in that case, we have a second option, which is to order the new adapters of fragments that are available at Twist for unlimited plug-and-play approach. So the way we do this is that we order several different fragments, the first one being a VHCH1 fragment, and then just standalone VHs, and 2 different types of FC fragment that could carry any types of mutation, whether that may be the beads mutation or additional FC engineering, with the CH1 or just the CH2, CH3 domain. And by using a direct ligation from the Twist fragment without the need for initial PCR amplification of any of those fragments, we can get endless possibilities of combination of VH and FC portions in 1+1 and long chain 2+1 formats. That's really efficient for us and gives us ease and speed of execution for our Beat 2.0 projects. Now, moving on to the example purification of a bispecific antibody I told you about. One more thing to know about the Beat technology is that it also encompasses a built-in purification strategy. As you can see, the CH3 domain in arm A is depicted in a different color. That's because this domain comes from IgG3, whereas arm B is fully based on IgG1. And because it's based on IgG3, it means that this arm A is enabled to bind protein A resin. And so if the homodimer of arm A is produced in your cell during the transfection at low level, this homodimer will not bind protein A. And once you elute it, it will actually elute in the flow through. Then the heterodimer binds protein A through one binding site in arm B. And so it will elute at a mild pH. And you will have to get to lower pH in order to elute residual B homodimers that carry 2 binding sites for protein A in the Fc. And with that technology, we have reached highly pure bispecific antibodies after a single step of protein A purification. You can see that in this reverse phase HPLC trace with MS identification, mass spectrometry identification of the different B2.0 species after a single step of protein A purification. We have produced the homodimers separately to know where they show up in this reverse phase chromatogram. And as you can see, we have not been able to find by mass spectrometry any trace of the A homodimer in this case, but we have found 98.3% purity for the heterodimer, that's the desired species, and only 1.7% of the B heterodimer carried over in this purification after a single step of protein A purification. Now with this B2.0 technology, I told you we can design and produce immune engager with diverse format and also tri-specific. And this is what we have done multiple times. And this is just a snapshot example where I'm showing one 1+1 B2.0 construct that carries 2 different binding arms for 2 different tumor antigen. And this was 98% pure by sec after a single step of protein A with almost 100 purity by mass spectrometry and yields that were comparable to standard monoclonal antibodies in the same setup. We were also able to produce 2+1 B2.0 that carry 2 times the same binder for a specific tumor antigen and 1 binder to another antigen. This was also produced with high yields and purities. And we have also produced a variety of tri-specific antibodies that carry 3 different binders, depicted as TAA1, 2, and 3 here. And this was also highly pure by size exclusion and mass spectrometry after a single step of protein A. And with that, I would like to thank all of the people who were involved in this work in discovery and in analytical development at Ichnos Sciences. And thank you for your attention. I'll be happy to take any questions.